I'm Peter Allen, and I'm the director of Google University, and I'd like to start first by acknowledging NGDU and MCS Events, who have really helped us get this event going. It's my honor to welcome our guest speaker, Karl Deisseroth, who will be speaking about cracking the neural code. Because he's such an honored guest, we have another honored guest to introduce him, John Hennessy, who's the president of Stanford. So I get to introduce John, and we decided not to make the line any longer than that. President Hennessy has been on the faculty at Stanford since 1977 in electrical engineering and computer science. He's a pioneer in computer architecture, and is currently or recently working on the architecture of high-performance computers. Very widely published, has received many awards, and is also on the board of directors at Google. Right, enough. So Stanford, in case you're not familiar, is a small college just up the road. Google University, just as for a second, helps Google employees develop cross-functional skills, including leadership, management, fundamental business and professional skills. And our School of Personal Growth, which is sponsoring this lecture, helps Googlers develop their full potential on all levels and has a particular interest in applied neuroscience. So that's really one of the reasons we wanted to have this happen. Please welcome John Hennessy.
So I think it's a time around the world where universities have to be thinking about attacking and going after the most ambitious, the biggest challenges we face. It was 50 years ago that Watson and Crick published their DNA paper, and since then, the work that's gone on DNA, on genomics and the human genome, and more recently on functional genomics, has revolutionized medicine. Not only laid the foundation for science, but changed the way we think about treating lots of diseases. And we're still in the infancy of that revolution. I think it's a time when before us stands the challenge of building the same scientific base for the human mind, so we understand the process of going from neurons to full cognition. Doing that will require lots of innovation and lots of new research. But I think one of the most important steps forward in doing this and building up that knowledge, which then will allow us to build the therapies that we really need to attack diseases of the brain, one of the most important steps is bringing together teams of scholars across the range of disciplines that will be necessary. Not only basic neuroscientists, but people who work in cognitive neuroscience, who work in psychiatry, who work in psychology, who work in computer science, who can begin to put together that model to crack the neural code. I think in order to do this, one needs some real breakthroughs in how you monitor, understand, and stimulate the human brain. And the talk you're going to hear from Karl today, I think, is one of the most important new breakthroughs we have for being able to monitor and stimulate human brain activity. Karl got his undergraduate degree at Harvard before seeing the light and coming to the West Coast. He did his MD, PhD at Stanford in our neuroscience program. He's currently a faculty member with a joint appointment in our new bioengineering department and in psychiatry. And most recently, he was winner of one of the very distinguished NIH Directors Pioneer Awards. This is some of the most exciting work I've seen going on in neuroscience. It's going to relay the whole map for the discipline. So I have to get out of the way and let Karl talk about it. Karl.
So thank you very much, John. And I have to say that the work we've done, it couldn't have been done anywhere else but Stanford. And I'll show you why that's true as we go along. But it's really an amazing place to work. I had my first tour of Google this morning, and also clearly a similar thing here. I was an early adopter of Google, so I'll take a little credit for that. But you've got a great thing going here as well, and it's wonderful to be here. I am in bioengineering and psychiatry both. I see patients 1 day a week. Most of the time I'm in my bioengineering laboratory, though. That's an unusual combination. You don't hear those in the same sentence too much, but I think the time is right for that. In psychiatry and in neurology, there's something wrong with the high-speed dynamics of the system, and we need better tools and better technology to probe and understand what's going on. But even beyond disease, clearly the brain does wonderful and mysterious things, and what we lack is technology that allows us to gain deep insight. So it all comes back ultimately to technology. The technology we've developed is called optogenetics, and in a way it has its roots back in some sort of visionary ideas that Francis Crick of Watson and Crick that you just heard about. He said what we need in neuroscience is we need a way to drive all the neurons of just one type in the brain and leave the other ones unaltered. And this is because the brain is it's like an orchestra, but with all the different instruments intermixed. And so you've got the bassoons and the piccolos and the harps all intermixed, and there's no way until very recently to really conduct this and to generate music, to control the different players individually. Crick foresaw that, and this was in a Scientific American article back in 1979. And I think in really an amazing turn of events, what's allowed us to make headway on this challenge is with tools from microbes, from algae, from single-celled algae, from ancient forms of bacteria that live in rare and fragile ecological niches. And I'll tell you about how that works. It's really an unforeseen turn of events that's really been uplifting. Now, come back to this issue as to why the brain is so difficult. We have within our brain many different functions that are controlled by many different elements, many different cell types. If you just think about one particular structure, the hypothalamus, this is a tiny little structure deep in the brain. If you zoom in, the scale bar here is 50 microns, 50 millionths of a meter. You see some red and green cells that are labeled here, all intermixed. Well, in the hypothalamus, look at all the different things that the hypothalamus does. It controls sleep, hunger, thirst, sex, growth, temperature, fear, social bonding, and actually many more. And all the different neurons, all the different cell types that do this are intermixed. So you can't stick in an electrode and just drive one. You'd get all of these going at once. Okay, so no one wants to be a teenager again, right? So that. And the red and the green ones here happen to be the ones for thirst and social bonding. What we need is a way to truly conduct the circuit. And as I alluded to, if you put in an electrode, you drive the cells you want to target and the cells you don't want to target. And this applies to basic science, figuring out what the brain's doing, but it also applies to treatments, to therapies. You'll get the therapeutic effect from one, but you'll get side effects from the other. The way the optical approach works is that we make just the cell type we want photosensitive, sensitive to light. And we can deliver the light with fiber optics, and we can control just the cell type we want and leave the other one unaltered. And so this is a large-scale effort. It's almost a Manhattan Project. It's how do you bring together an immense diversity of ecological tools, of computer science tools, of optics, of behavior, of psychiatry and neurology. How do you bring these all together in a common enterprise to make this sort of effort work? And there are three components to it. One is bioprospecting, mining evolutionary and ecological diversity for the tools that we need, and nature has made wonderful things. Reverse engineering, applying the control tools we have and the readout tools we have to try to infer what sort of computations, what sort of motifs are present in the brain. And finally, of course, this is relevant to just the basic science. How does it work? But you'll see how important the implications are for disease as well. So here they are. These are our unlikely allies. This is basically pond scum. This is Chlamydomonas reinhardtii. It's a plant. It's a single-celled algae, and it has a chloroplast. It photosynthesizes, uses light to generate energy, and it has flagella, so it swims around in freshwater ponds. So it uses light for its own purposes. It doesn't care about the brain. It doesn't care about the neural code, but it uses light for its own purposes, and I'll show you how we can co-opt that. This is an archaebacterium, a very, very ancient form of life. These forms of life they've been sort of shouldered out. They live in only very difficult, challenging niches in the world. They live in very high temperature environments, hot springs, or they'll live in very severe salt conditions, as this one does. And they use light because it's one of the few resources they have around, and they use light in order to do things like move ions around, and that turns out to be very useful for us as well. And the reason is this: they make proteins. They make small nanoscale devices that basically act as antennas that receive light and transduce the light signal into ion flow. Here's sodium and calcium ions. The one from pond scum is called CHR2, or channel rhodopsin 2. It's a single protein that inserts into the surface of a cell. This represents the membrane, the surface membrane of a cell. And it acts as an antenna for light, and it converts the light signal into the net entry of positive ions into the cell. It does this for its own reasons. It does this so it can learn to turn toward light. As being a plant, it has to do that. Well, this happens to be neural code for on. If you have a cell and you drive positive ions into the cell, this is the currency of information flow in the brain, and it means on. It means active. So that's fortunate for us. The archaeobacterium acts as an antenna for yellow light. It makes another protein that responds to yellow light, and it pumps in a negative ion, an anion, chloride. Well, this happens to be neural code for off. And so we're able to co-opt these tools for our own purposes. Here's a close-up of what channel rhodopsin 2's source, the algae, looks like. It's got a chloroplast. This is how it photosynthesizes. It makes light. It makes energy from light, and it has an eye spot to detect light, and it has flagella that it uses to swim around. Now, it's pretty cute. It rotates on its long axis when it locomotes, and so its eye spot spins around. So if light's coming from one side, it's got an oscillating light signal that it needs to make sense of. And it doesn't have a brain to process that. It's a single cell. And so it needs to be very efficient, and you'll see this theme show up again and again. These microbes have to do everything all in one package, and they have to be efficient. And so what it does is it makes a very fast light sensor that transduces the signal into ion flow. Now, we used genetic tricks. We took this light-activated channel, and we put it into neurons, neurons from the brain. Here, neurons from a rat brain. And the scale bar here is about 50 microns, 50 millionths of a meter. And what you see here are the outlines of 3 neurons. We put this into these cells, and we tagged it with a yellow fluorescent protein. The Nobel Prize was just given for a green fluorescent protein a few weeks ago. This was given to Marty Chalfie and Roger Chen. This is just a static imaging tool. We tagged this channel with a fluorescent protein so we can see where it goes in the cell. And it goes to the surface, so it's right on the membrane, right where we want it. And this was the most amazing thing. We couldn't believe this when we tested it out. We give these millisecond scale pulses of blue light, as shown here by the blue dashes, and these sawtooth deflections, these are what are called action potentials, or spikes. This is the currency of information flow in the brain. So with pulses of blue light, we can now start to speak the language of neurons. And because it's a single gene, we can now target it to different cells, and we can now start to control them with light. Now, can we get this working in freely moving animals? Well, what we have here is a mammal, a freely moving mammal with a fiber optic. It's all externalized here so you can see it. This can be internalized. These fiber optics that we can use 50 microns, 100 microns diameter fiber optics. We can target any area of the brain. We can go deep, superficial. We can have multiple of these fibers. And so I want to show you a movie. This is a movie of a mouse where we put the fiber optic in the right side of its brain. And you may know the right side of the brain controls the left side of the body, controls movement toward the left side of the world. And in the surface of the brain, there's a 6-layered neural network. It's called the neocortex. 6 layers, one on top of the other. We used a genetic trick to just label layer 5. And what you'll see, if this movie plays, you'll see, now why is it not playing?
We've got no here.
Oh, wonderful. You'll see this animal circling, and I'll play it again. We turn off the light here, and it stops. Okay? So we made this animal really want to turn. I'm going to play it again so you can see it again from the beginning. You can't see the fiber optic because it's so light and thin. You'll see a little blue light reflectance from the skull when the light goes on. Let's play this again. So just in layer 5, you can see he's just exploring, and right about here you'll see the little blue light turn on, and he'll start to really want to turn left. You can see he's normal locomotion, doesn't appear distressed, and he stops right here when we turn it off. Goes right back to exploring. So that's, on one level, it's a little scary, right? I mean, here's a freely moving animal that we've induced to really want to turn left. And this is something you could never do with an electrode, just driving that layer 5 here at 20 hertz, 20 times a second. And because it's a fiber, we can target any area of the brain. And this is something that we do quite routinely. So I'm going to come back to this issue again. What are the ethical implications? What are the philosophical implications? But also, what are the positive implications, the medical aspects and the basic science aspects? Now, this is only one axis of control. This is using blue light to drive things, to excite things. But I can tell you this is just the tip of the iceberg, okay? What's illustrated here is sort of an artist's rendition of the kind of thing we can do. We can search this sequence of letters here as an amino acid sequence of a protein. We use search tools, which you are very good at here. We use search tools to go look through genomic information and find novel tools. We can predict from the sequence whether they'll respond to blue light, to yellow light, to green light, and we're starting to build a palette of different channels of control that can all work together at the same time. And so then we can truly start to play the role of a conductor, drive different cells in synchrony, out of synchrony, as we wish. And this is a new kind of algae called Volvox. I'll show you a picture of that shortly. The amazing thing is that life in all its complexity, bacteria, archaea, these are the archaebacteria, and eukaryotes, we live here. This is a relatively small, unimportant branch. All of these use external signals for information and for energy. But the microbes do it best. They do it most efficiently because they're small. They don't have much space for different genes, so they have to do everything in one gene. They have to be compact. And maybe even more importantly than that, they're incredibly diverse. And so in every niche, every spot where life can exist, there is life. Each spot has its own source of energy, its own unique set of environmental information. And I can guarantee for virtually every kind of energy, every kind of information out there, whether it be magnetism, pH, light, there's a microbe out there that spent millions of years designing a beautiful antenna for that source of information. And so the goal is to actually extend this and expand this to sound, to magnetism, to other wavelengths of light. And this is a form of bioprospecting, but with a clear, focused goal to develop the palette of tools we need to truly start to speak the language that we need to understand and control the brain. Now, I mentioned green fluorescent protein, and I mentioned this is just a fluorescent protein. This is something we use as a tool in the lab. It's been around for about 15 years. We didn't discover it. It was discovered by Marty Chalfie at Columbia. And as I said, he just got the Nobel Prize for that. But it was shared with Roger Chen at San Diego. And why was it shared? Roger Chen made red-shifted versions of it. He made yellow fluorescent protein. He made red fluorescent protein. Now, these are not control tools like the tools from the microbes I'm telling you about. These are just anatomical things. They let you see things. They're just static fluorescent things that let you track and trace where cells are. But why did Roger Chen share in the Nobel Prize just for making red-shifted versions of it? That doesn't seem that much of a big advance. Well, actually it is, because the power of having this palette now gives you combinatorial imaging. So you can now see, where do things overlap? Where do they intersect? When do they share properties? When do they not share properties? And that has turned out to be such a big advance that it was certainly worthy of the recognition that he got. So with channelrhodopsin 2, this source of control, remember, fundamentally different from this in that it's control now, can we also develop shifted versions of this and start to actually play things in complex patterns? Well, the Neutronomonas ferrionis halorhodopsin starts to get us down that path. And you can see it responds to yellow light. It pumps in chloride ions. And where does it come from? It comes from a fascinating geological set of structures. These are salt lakes in the Sahara Desert in Egypt. And these are constantly forming and evaporating. They're fusing and disappearing. And they achieve very high salt concentrations, 2 to 3 molar. So this is extremely concentrated. Not much can live there except for these little archaebacteria. And they can live there because they've evolved these pumps. They evolved these pumps to control their ion gradients and to use their ion gradients to generate energy. So we did the same thing. We took the halorhodopsin, the chloride ion pump, and we put it into neurons. In fact, we even put into neurons where we had the blue light activator, the channel rhodopsin. We put them in the same cells at the same time. And then look at these little sawtooth deflections. We can drive these action potentials, these spikes of blue light pulses, and we can shut them off with yellow light, because remember, negative ions coming in, that's neural code for off. So bidirectional control, different channels. Well, now you say, well, that's one color on, one color off. We need 2 colors on, right? We also need that because we'd like to positively drive cells in synchrony or out of sync. And we've taken steps toward that as well. We found this Volvox channel rhodopsin. And this is a fascinating little algae. It's actually not a single-celled algae, it's multicellular. And it comes in these spheres. It's more of a social algae, you know, they live together, they cooperate. It's called the fierce roller because it's spherical, and it has sort of a rotational form of locomotion. But it turns out, and we found this in looking through, this is just an illustration of what the genes look like. This is in the amino acid sequence of the genes, what it looks like. And we went in and we found this one. It's called VCHR1, Volvox channel rhodopsin, and it had some subtle differences compared to the other ones that we'd seen. And in fact, you could predict that it would be red shifted. And indeed it was. This is an action spectrum. We call this an action spectrum. This is the wavelength of light that works to excite the channel. And blue light activates the older one, the channel rhodopsin 2. You can see 470 nanometers, that's blue. Well, out and around green, that's about 525 nanometers. And so you can see, and we can use this tail out here of the VCHR1 action spectrum, the Volvox channel rhodopsin spectrum. We can use this tail out here to drive it that doesn't touch the channel rhodopsin 2 itself. And we put this into neurons, and we used yellow light out on that tail. And look at this. We can drive these spikes now with yellow light, 589 nanometers. This is a wavelength that doesn't touch the other one, the earlier tool, at all. So now we've got multiple channels of excitation as well as excitation inhibition, starting to build the palette of tools. And this is all just light, okay? Not even starting to get into the other forms of energy, the other modalities that are relevant. Now, but looking at them all together, so now we've got the blue light activator, the yellow light inhibitor, the green light activator, and these share a number of very important properties that make them very useful for translation to use in mammals. And they're single component. This is incredibly important for delivering them, for technical reasons, to get them to work well. We can target them to individual cell types. We can tune them, wavelength tune them. We can do combinatorial control. The fact they respond to red-shifted light allows us, red-shifted light penetrates deeper into tissue, so we can access deep structures that way. They work in behaving mammals. They're fast, millisecond scale. This is crucial. The brain operates on the millisecond scale. So without that, it's hopeless. This is why drugs, pharmacology, they have so many side effects. They usually don't have the effect you want. It's because they're not operating on the appropriate time scale. There's no dark activity. Nothing happens until you turn on the light. The light power that they use, 1 to 10 milliwatts per square millimeter. This is tolerated, and this is because they come from biology. And so it's designed, of course, to be compatible with life. These are made and used by biology. And you don't have to add chemicals or other cofactors to make them work. So a lot of very important properties. And again, we couldn't have done it without nature. So this is only the tip of the iceberg. We're expanding the search now, scaling it up, looking not just for light, but for antenna and these nanoscale antennas that will be able to respond to other modalities of energy and information. So that's the sort of the first leg of it, the bioprospecting leg, and you can see the sort of progress that's been made. Now, of course, the next step is to try to use fast control tools and fast readout tools and try to infer what neural circuits are doing. Now, let me pose a really fascinating question for you. So this is the brain. This wrinkled structure that you see when you look at the brain is actually only part of the brain. It's called the neocortex. And this is this 6-layered structure that sort of sits like a coating on top of the brain. All the wiring is inside. The 6-layered set of layers that cover the brain, these are layers of cells, of cell bodies, and they send out axons or wires that connect them to other cell types. That's all in the middle. But the coating is on the outside. The brain is specialized. It does different things in different regions. This is the back of the brain. This is the occipital lobe. It's specialized for vision. And so all visual sensory input ends up here, gets processed. This is the temporal lobe. This is involved in auditory processing, but also in mood and in memory. This is the parietal lobe. This is involved in calculation and spatial rotations and integrating sensation. This is the frontal lobe. This is where planning, executive function, motivation all reside, where motor output resides. Totally different roles. Here's the paradox. You go in and look at these with a microscope, and they look exactly the same. Okay? You look at the neocortex here for visual input, and you look at the neocortex here for planning motor output. It looks almost exactly the same. Subtle variations in the thickness of the layers, but the players are all there. It's a 6-layered structure, same cell types, same wiring. Same thing here. And this mystery is also a clue. And it's sort of analogous to what John alluded to with the double helix. It's the same kind of commonality that you see in the use of DNA and you see in the use of the double helix-based genetic code. It's the same code in virtually all forms of life, some subtle variations in some forms. Of course, incredible diversity in life, but clearly there's a common theme, a common motif that underlies how the genetic code works. It's got to be a similar thing here. Different areas of cortex do totally different things, but the same fundamental structure. There's got to be an underlying motif, an underlying dynamical code that is accessible and understandable. Here's an illustration of what this looks like on a slightly finer scale. These are the 6 layers of cortex. 1, 2, 3, 4, 5, 6. Information comes in from a structure called the thalamus. It comes in here, it hits layer 4, hits layer 5, gets sent up to the superficial layers near the top, gets sent down to the deep layers near the bottom, and then it gets output back to the thalamus where motor output behavior is expressed. But it looks the same basic structure, same basic cell types, no matter where you look. So the schematic, the microscopy doesn't tell us enough. It tells us the players, but it doesn't tell us what the circuit's really doing. For that, we need dynamics. We need to play in complex patterns of stimulation, and we need to read out complex patterns of output. And that's a black box, okay? So what there is there's a black box in there. We don't know what it's doing. We might be able to turn this into a gray box relatively soon, have some rough models that we can start to fit the data to. But the key thing is to have fast play-in, and this is another illustration here, driving spikes with blue light pulses, knocking them out with yellow light. This millisecond scale control we can now use to play in complex patterns of activity. At Stanford, we and our colleagues are developing fast readouts as well, electrode-based, imaging-based, and behavior-based to get the readouts to see what the circuit's doing. And the goal is to turn the black box into a gray box and then finally to understand what the underlying dynamical motifs are that are present in the brain. This is an illustration of how this can work. What's shown here, this is an entire rodent brain, so you can get a sense from the scale. It's sort of on the centimeters scale. We can play in complex patterns of activity, just as you would do if you're conducting an orchestra, playing in different colors of light, funneling through fiber optics. We're getting increasingly precise control of the spatial pattern of activity. We can get readouts from the structure. We can do electrical recording from candidate downstream structures. We can do imaging to get fast readouts. And most importantly, we really know what it means because we can do behavior as well. We can have freely moving animals and we can see what the real consequences on the behaving animal are. So that's the second leg of this, which is the reverse engineering leg. And the final leg that I think is for me one of the, not that the other aspects are not interesting, but one of the most satisfying for me is the potential for medical translation. And here's the problem we have with the brain is the paradigm of neurotransmitter soup, or the chemical imbalance. And I'm sure you've all heard of this. When you have someone who's depressed, does that person not have enough serotonin? Or if someone is hearing voices, do they have too much dopamine? Okay, and this treats the brain as if it were just a soup of chemicals, without millisecond precision, without complex structure. And this is why medications in psychiatry and neurology, they have serious side effects. They help some people, absolutely, but they also don't help many more people. And they don't speak the language of the brain. Here's epidemiology, which I think is underappreciated. These are numbers from the United Nations. The World Health Organization has quantified the fraction of disability worldwide or in the Americas, similar in both distributions, the fraction of disability worldwide caused by neurological and psychiatric disease. And you can see it's the red pie slices. It's enormous. These diseases are often fatal. They're hard to treat. They're chronic, and we just don't understand them. We just don't have good models, and we just don't have the technology to generate good models. And this is targeting the hypothalamus is actually an interesting paradigm to show you that we can actually make headway on medical applications, medical control. So I showed you this image before. This is the hypothalamus, and you've got two different kinds of neurons all intermixed. Well, we did a study to see if we could control the neurons that were involved in sleep regulation. Remember, that was one of those things that the hypothalamus does. There's a disorder called narcolepsy, which you might have heard about. It's studied at Stanford, outstanding and pioneering work done there. There are neurons embedded within the hypothalamus called the hypocretin neurons, also known as the orexin neurons, and they're thought to play a role in narcolepsy. When you lack these cells, whether you're a human being or actually animals that lack the receptor for the protein made by these cells, you have the tendency to fall asleep. Particularly when you get excited, you fall asleep. And this is a serious debilitating disorder. Well, we went in, we put the channel rhodopsin, the blue light activated probe, just into the hypocretin neurons that are involved in wake. And this is what it looks like. We just put a fiber optic down. We used a couple 100 micron diameter fiber optic, went all the way down to the hypothalamus. We did simultaneous EEG to look at the brain waves. And we had a freely moving mouse. And what you'll see here, this is an EEG. Just focus on this one here. This is an EEG of an animal. And here's where we give blue light stimulation at 20 hertz. And you can see a few seconds later, time scale here, 10 seconds. This starts to change. Now, this may not look too different to you, this little stretch here compared to here. But if you look at enough of these and you quantify this is lower in amplitude. And look, the animal's waking up. This is the muscle graph indicating that it's starting to move around. So we can actually drive awakening. We can drive this complex behavioral state transition just using blue light, just by driving the hypocretin neurons that are intermixed with all these other cell types in the hypothalamus that do all these different things. And so this has very important implications for narcolepsy, but also just as a proof of principle that we can now start to apply this to medical problems. Another major class of medical translation has to do with deep brain stimulation. This is where you put an electrode. You actually place an electrode within the brain. There's a wire running down the neck, and there's a battery and a controller under the clavicle in the chest wall, just like a pacemaker battery would be. And this is used in Parkinson's disease. It's also used in depression. And although it doesn't work for everybody, and although it causes some side effects, some really interesting things are seen. When you put this into an area of the brain that's probably involved in mood, you can see as soon as the electrode turns on and you drive it at 130 hertz, 130 times a second, depressed patients report things like this: sudden calmness or lightness, disappearance of the void, this classic depression symptoms, emptiness, void, goes away in many patients. Not all, or even most, but some. When the electrode goes on, the void goes away. Now think about that, because this is, as you well know, a lot of these things, the low energy, loss of hope, void, emptiness, these are hard things to express. They're hard things to quantify, but they're real, they're fatal, and they underlie an immense source of disability worldwide. And this actually starts to show us that these are real, tangible things, that the biology is there, it's accessible. It's not good enough with the electrode, although it's wonderful work, but it's not good enough. It doesn't help enough patients quickly enough, and it can have side effects. It's dose limited, but this is really, really important. It's an important proof of principle that we're headed down the right path. Heightened awareness, increased interest, connectedness, brightening of the room, sharpening of visual details. I've talked to Helen Mayberg, who's done this work. She says patients start wanting to clean out their garage or plan a trip to Italy, So, of course, within reason, you don't want to do too much of this. But think about this. It's quite profound. 130 hertz, well, that can't be the neural code for happiness, right? That's too simple. And indeed it is, but it shows that major psychiatric or neurological symptoms are accessible, that maybe some of the problems have to do with activity not getting through a bottleneck, and the bottleneck can be tweaked in a relatively a simple fashion like this. Vagus nerve stimulation. This is another way of getting to it. The vagus nerve is a nerve that runs from the brain down the neck to the rest of the body. There are also nerves that run up through the vagus nerve from the body back to the brain. So it's like a superhighway. You can actually access the brain through the vagus nerve without actually doing brain surgery. And so this has been used for epilepsy and depression. You put a little cuff around the vagus nerve, battery pack in the chest wall, but you have a lot of side effects again, and this is because electrodes are crude and nonspecific. Look at the rate of side effects, voice alteration. It starts to sound strangulated when the device goes on. That's because you're activating all the throat neurons, the laryngeal neurons. Cough, difficulty breathing, difficulty swallowing, neck pain, okay? So it can work, but side effect limited because the electrodes are imprecise. These are huge disorders. Everything that's in principle accessible, treatable with electrical stimulation. You look at the list here: depression, chronic pain, obesity, OCD, addiction, Alzheimer's, Parkinson's. Very common, very costly. We know where the foci of these disorders are. We know approximately where to go, but the electrodes are not quite good enough and don't quite take us there. And I want to touch on, getting toward the end now, I want to touch on recent work from my lab on Parkinson's disease. So here's two individuals with Parkinson's disease. It's a very serious disorder. More than 1 million Americans have it. It's got a chronic course. It's ultimately fatal in many patients. One of the key symptoms is slowing down. You can't move quickly. Eventually your gait just becomes sort of a tiny stepped shuffling gait. Serious tremor, sort of a 5 hertz tremor. Progresses to dementia, depression, and death. Very, very serious disease. What happens is there's a set of neurons that live in a part of the brain that's deep called the substantia nigra, because with a particular stain they look black. And these are the dopamine neurons in the brain, and they get lost in Parkinson's disease. They disappear. And that gives us we know that a particular cell type is missing or not working well, but it doesn't tell us yet exactly how to intervene. And we don't know. If we did know, we'd know how to treat this. We don't know exactly how to intervene to help most patients. Now, deep brain stimulation is done for Parkinson's disease. You can put the electrode in downstream brain structure, drive it at 130 hertz, and hope for the best. Well, that works, actually. In a significant fraction of patients, you get recovery from the symptoms of Parkinson's disease. The problem is it doesn't work for everybody. It stops working after a while, and it is side effect limited. And so again, can we take this to the next level? Can we provide, instead of doing a crude 130 hertz stimulation of the brain, can we start to actually fix the dynamics of neural circuits in a coherent way? And here's an illustration. We actually, I use the word deconstruction here. This is really what we're doing. We target the light-sensitive proteins to all the different elements of the circuit sequentially. We march through them one by one, and we find the right tweak, the right knob to turn that corrects the disorder. Here's the path of a mouse before, during, and after the optical treatment that we did when we found the right circuit element. And this has taken over about a minute. You can see that, remember, bradykinesia, slow movement, that's one of the core symptoms of Parkinson's. Barely moves at all, 10 centimeters only, before we go in and we drive the right circuit element. We found the right one after a lot of searching. A lot of our early efforts didn't work. But it locomotes quite readily. Once we turn on the light, then we turn the light off, and it's back to the Parkinsonian state. And so this, for us, was a really wonderful validation of the basic principle of deconstructing circuits, not just for basic science, but also for medical translation. And again, the goal really is to march through the disease circuitry, whether it be depression, whether it be Parkinson's, and understand how the dynamics are off and how they can be tweaked and tuned to correct the disorder. So these three legs, the bioprospecting, the reverse engineering, and the medical translation, you can see how they stand together, but how broad they are. On different fronts, you need genomic experts, you need people who are very good with search, with DNA, very good microbial biology. This is a very important engineering, computer science, electrical engineering concept, medical translation, physicians, people who are familiar with disease models. And this sort of broad effort, I think, is the kind of thing that can help sort of really unify a large organization and toward a common goal. For us, it's a very exciting moment. Now, there are ethical implications as well. You know, anytime technology takes a big step, there should be serious philosophical consideration of what's really going on. What can we control? Well, getting back to the hypothalamus, this is where all our priorities are, okay? Where all our needs, our desires are. That tells us how much we want of something, what we want, and how much we want it. And that being accessible now, that's a serious issue. Now, as you can see, as nice as the fiber optics are, this is not the kind of thing that can be projected from 1,000 miles away. Okay, it's not the level of a weapon, and nor do we intend it to be. It's not really suitable for that. But it has, in addition to that sort of level of concern, in addition to the scientific issues we can address and the potential ethical concerns, there are philosophical concerns. You know, this is, if what we really want, if our needs are really tunable by a particular cell that's present at a certain level in a particular structure of the brain, does that have implications for understanding what it really means to want something, what desires are, what priorities are? And these things, I think, have to be considered along the way. And these are, again, we have wonderful colleagues at Stanford who are very good, along with us communicators on this issue. Last slide, two final thoughts. And this, to me, this is what I feel most passionate about. These tools come from basic science, from people who just studied for decades these organisms because they thought they were interesting, without a thought to application, without a thought to medicine, much less neurology or psychiatry, okay? So these were, we didn't discover these organisms. We discovered the use of them for neuroscience, for application to science. But without the decades of work by the people who had studied these, we wouldn't have made much progress. And so I think it's an immensely important testament to the value of basic science that be allowed to proceed, to be encouraged to proceed. This is the other point, ecological diversity. Many of these come from rare or fragile ecological niches. There'd be no particular reason to worry about a salt lake in Egypt, right? Except on general principles. And so I think what comes out of this, knowing that tools arise from rare and fragile things, I think it points us to what can we do to preserve the rare and the fragile and the weak. And so I just want to conclude there, and thank you all for your attention. Yeah, I'd be happy to take any questions and chat with people. Feel free to email me, Yes. Yeah, it's a great question. I didn't have time. Yeah, I will repeat it. It's a great question. The question was, how do we target these probes to specific cells in the brain? I didn't have time to touch on that. What we use is we capitalize on the fact that different cell types in the brain have different jobs, and they make different proteins for those jobs. And so different cells will make protein A and not protein B. They do that using DNA. There are little bits of DNA called promoters which govern expression of protein A only in that cell type. A lot of these are known, and we know more and more of them all the time. So we take that little bit of DNA called a promoter, which this cell uses to make sure only protein A and not B is expressed in the cell. We take that little bit of DNA called a promoter, and we use a genetic trick. We attach it to the DNA that encodes for our light-activated protein, and we put that whole complex back into the brain. So that whole complex goes into many different cells. In fact, it goes into all the cells. But the promoter that governs production of things is only active in the particular cell type that needs to make protein A. So that's a trick. We capitalize on the fact that cells already have genetic tools to make sure proteins only get turned on in one cell type and not another cell type. Yes?
Diving into that a little deeper, how do you said you put that back into the brain. What is the mechanism for putting it back into the brain?
That's right. So once we've put these two bits of DNA together, how do we get them back into the brain? This actually is the easiest part of the whole thing. It sounds like the hardest, but at this point, this is easy. And we do this with things called AAVs. These are called adeno-associated viruses. We all have them. We expose them all the time. These are part of normal cold viruses, but we tweak them so they no longer can cause cold symptoms and they no longer can reproduce. And we put the DNA in there. These are now widely used in people to deliver genes. There was just a study in the Lancet published using this kind of system to deliver a different kind of gene for human beings with Parkinson's disease. They're very safe. They're well tolerated. It's something to be careful of when thinking about translation to people, but it's being done. And actually, this is right now it's the least difficult part of the whole thing. Yes.
So what can we do to help you?
What can we do with what?
To help you.
Well, as I've said, the people here in so many ways touch on different aspects of this. We do need better ways to search genomes, and we need better ways to reverse engineer circuits. We need outstanding computer science, and we need outstanding smart people who like searching through things. And so there are very natural synergies. Yes.
So the nematode wiring of the neurons has been known for a while, but I understand it's hard to get from that to the functional, how it actually works.
Yeah.
And it seems like what you have here is the missing piece to actually walk through and figure out what the neurons do. Has anyone looked at applying this to simpler organisms?
Yeah, so this is a wonderful question. So the nematode worm, the actual wiring has been known for a long time. And this is because it has only 302 neurons. And so with serial electron microscopy, much as you would reverse engineer a smart card or computer chip, with electron microscopy, the entire wiring of this little worm, this nematode, was discovered actually decades ago. But although it was done decades ago, we still really have essentially no understanding of how even that nervous system works as a unified whole. And the reason is we lack specific control and we lack dynamics. We lack the ability to probe and modulate and read out dynamics. So you're exactly right. What was missing was the element of fast control. Not that you don't need the wiring diagram. You need that. The missing piece is adding in the fast control. Yes?
I think you made a convincing case for why the pharmaceutical approach to treating mental disorders is not particularly effective. Could you leap forward several decades and imagine what the clinical applications of this might look like?
So what are the clinical applications? And I think, we're already starting to touch on those, and those are coming in the form of understanding. What are the clinical applications? We're now starting to understand what are the circuit elements that are going wrong in Parkinson's disease, and there's a lot more to learn. We're just scratching the surface. But just understanding will help us do things like design better drugs, design better non-invasive interventions. We have non-invasive interventions that don't require even fiber optics, even something as thin as a fiber optic. We just don't know how to use them well because we don't understand. We don't understand really what's wrong with the circuit. And it's very hard to do high throughput search of a circuit in a human being who's sitting before you in the office to find out what's going wrong with their circuit and then to correct it. So the understanding from the animal models will help us use even other tools better in people. In the very long run, some of these actual tools might be translated to people, but I see that as a minor aspect of the whole thing. I think the real goal is understanding and design of better treatments, which will come very quickly, probably on the 5 to 10 year time scale. And so we're looking forward to that. Yes?
Where can we read more information?
Okay, yeah, where can we read more information? First, we have a website, and it's optogenetics.org, so optogenetics.org, all one word. And there's links to publications, images, methods, and press there. And you can email me also. Yes?
A lot of what the brain does happens in networks and with feedback from many places at once. So I was curious, are there applications that you're working on for activating multiple regions at once and inferring the architecture underneath?
Yes. So bringing up the very important issue of brain feedback, of the way different regions of the brain work together, one influencing another. And this is very important. Actually, a lot of diseases may arise from feedback, from overactive feedback, effectively getting stuck in a loop that needs to be intersected, and that can be done even briefly. A brief intervention could knock you out of a vicious cycle that at least for a while. And so understanding those feedback loops is probably important in depression. It may well be important in Parkinson's. And you may not need multiple sites. You may just need to intersect at one point to break the loop at one point in order to affect a cure. That said, there are many reasons to seek control of multiple different regions of the brain at once. Probably different areas of the brain communicate better when they're in sync, and I mean on the millisecond scale here. So this is not just generally revving up two different regions of the brain. This means on the millisecond scale making sure they're in synchrony. This is probably how the brain unifies different parts of an object into a unitary whole. So I stand here, there's sound coming from me, there are photons coming from me, but you still see me as a unitary entity. And why is that? Because the different neurons in your brain that are responding to the different aspects of that entity, they're probably all in synchrony on the millisecond scale, and that's creating an object, a single unitary object. That is crucial to understanding how the brain works and how that goes wrong in some diseases like autism. And to do that, we're developing fiber bundles and multiple different fibers that can access different areas of the brain at the same time and drive them in or out of synchrony. Yes.
I understand that your interest is primarily in understanding the brain, which I sympathize with. But I'm curious, looking at this, whether you think that you could use this technique for anesthesia or pain control, possibly peripheral.
Yeah. Question is pain control or anesthesia. This is absolutely an application, with the key reason being, with electrodes, it's very hard to turn off. Okay, you can definitely drive things, and in some cases, you can overdrive them so hard that the neuron becomes exhausted and no longer works as well. But that's difficult. It has side effects, doesn't always work, and certainly doesn't spare the other cells around, so creating problems. And this is why pain control is so hard. In the spinal cord, where the pain pathways go, that's where motor control goes also, and you don't want to paralyze people. This has clear applications for pain because we can truly turn off using the chloride pump, and we can truly turn off just in targeted cells. And so we're looking at applications for that. The pain pathways are known. The promoters for the pain pathways are known. And so this is absolutely a possible application. All right, well, thank you very much. It's a pleasure to be here.